It's amazing to hear whenever we get to summarize these things. It's wonderful to hear. I just want to give everyone a sort of a primer on what he's saying when he says R or K. For the NIH, an R award, literally R stands for research, and they're numbered by the types of awards they are, and they're considered to be prestigious and recognize independent scholarship, and they want to fund you for something that's deemed worthy by your peers. K is supposed to stand for career, as far as I understood it. And I guess the NIH needs to go back to school. So when we talk about somebody who has a K award, it's a career development award for a junior person, and R is a research award. So that's what these different types of awards are. So now it's my great pleasure to introduce our next speaker. It's Sonia Kupfer, who is an associate professor of medicine, recently promoted, very well deserved, and also the director of our gastrointestinal cancer risk and prevention clinic. She's become an international authority on hereditary cancers and prevention of cancer, and has done some really groundbreaking work related to understanding the genetics of these, as well as metabolism of vitamin D related to some of this. And she's going to tell you how GERP has supported her.
Great. Thanks so much, and it's really wonderful to see a lot of familiar faces, some people I've known since I was in high school, but also a lot of new faces, so thank you so much for coming. And I'm going on 20 years since I started medical school at the University of Chicago, but it's the shoulders of giants like Dr. Kersner, but also Dr. Chang and Dr. Rubin that we really stand on and have really paved the way for us to do the things that we do. So similar to Dr. Rubin, my motivation is that I had a very close family member who we lost to colorectal cancer. So that's really the motivation behind why I do what I do. But what I'm interested in is understanding how your genes interact with your environment and your lifestyle to create health and disease, and in my particular case, to understand why people are at risk for cancer. And of course, we don't all look like this cartoon figure on this slide. We're a very diverse group of individuals, and so our cancer risk is potentially very different because our genes are different and because our exposures, our environment, and our lifestyle are different. And so this really forms the foundation of the research that I do in a lab. So I'm a physician scientist, which means that 50% of my time I'm in the clinic seeing patients, and 50% of the time I am in the lab, and 50% of the time I am with my family, and 50% of the time I'm writing grants, so you can see how this goes. But the idea is that I really want to understand why a particular individual gets cancer, and how we can prevent that, and if cancer were to develop, how we can better treat that cancer related to that individual. So these are some scientific slides, but hopefully you can appreciate this was really a breakthrough in how to study individual responses in the colon and in the GI tract as a whole. So these are called organoids, or mini-guts, and essentially what I do is I take biopsy samples from a colonoscopy that I'm doing every Thursday, and we are able to isolate stem cells from the colon, and we put them in a petri dish with a lot of media and growth factors, and these little stem cells in their little homes, they create these little mini guts, or these three-dimensional organoids of the colon, and you can get them from any person that comes in for a colonoscopy. And the nice thing there is that I can treat them with anything I like. So I've been very interested in understanding how individuals respond to vitamin D, because we know that vitamin D can prevent colon cancer. I'm also interested in how they respond to aspirin, because aspirin can prevent colon cancer. But I can also treat them with things like bile acids, which in some cases can cause colon cancer, or the microbes that live in our GI tract, or anything that I'm interested in, inflammation. And so by doing this, we can treat them with the treatment and also with a control, and in a very controlled setting understand how the colon is responding to the environment. And so these are images of our little organoids. They're our little babies, and we watch them grow and become three-dimensional, and then we look at them under the microscope and we treat them. This works. It's a little video of an organoid forming. There you go. That took a lot of work to do that. And so my hope is that through doing this, I can really understand inter-individual, and in particular, how different populations respond, because we know that certain populations, unfortunately, have much higher risks of colon cancer in particular, like African Americans. But also there are some individuals who have genetic mutations that lead to GI cancers. And so we can use these organoids to help prevent cancer, as I mentioned, to help treat cancer better in a very personalized way should it develop, but also to understand why certain preventative measures work. And so it's really been through GIRF's support that I've been able to eventually now get 2 NIH grants. I had a K award, but I've also now had 2 R awards, and it's really because of you that I've been able to do that. So thank you very much.
Okay, so we're going to ask you how to clone a colon when we're done. Our next speaker is Dejan Michik, and Dr. Michik is an assistant professor of medicine, one of our junior faculty. He's in his 2nd year now as a faculty member. He has a unique training background. He worked with us in his fellowship and worked with me learning IBD, but then he went on for a year at Northwestern and worked in the nutrition area and did some advanced nutrition and learned some advanced procedures to investigate the small bowel with scopes that go all the way through your entire small intestine, which is a very long scope. And now he's back focusing on some of these areas, and he's going to tell you a little bit about the work that GIRF is supporting for him.